Okay, this time we're taking a look at the phylum Porifera, which are the living sponges. So when I say living sponges, I'm kind of differentiating between your kitchen sponge, which is made out of rubber, and real sponges. And they are sessile filter feeders. So we're gonna start off looking at their lifestyle and their external morphology for the most part, or their macro morphology, I guess you could say. And so sessile is a term that refers to being attached. So they just stay in one place. They attach and they stay in one place. And they sit there and filter feed small food particles from the water. Now I want to remind you that sponges, or Phylum Porifera, is the very first phylum in the animal kingdom. So they have very close evolutionary relationship with the choanoflagellates in the kingdom Protista, in other words, protozoans known as choanoflagellates. And sponges are made up of cells that look very much like, or almost exactly like, the choanoflagellate cells that make up colonial choanoflagellates. And these choanoflagellates, some species are sessile filter feeders also, but they're not considered multicellular. Sponges are considered multicellular because they have a number of different specialized cell types. But let's get back to their way of life here. So they're sessile filter feeders, and what they do is they pump water through themselves. And so they bring water in through what are known as in-current pores. So the entire lining of their body has holes in it. That's where the term porifera comes from, pores. So there are all kinds of in-current pores, and water's coming in through the in-current pores, and it's coming into a space inside the sponge called a spongocoel. That part of the word seal refers to a cavity. So this is the cavity of the sponge, spongocoel, right? Just like a coelom is that second body cavity other than the digestive cavity that we were looking at in embryonic development in Kingdom Animalia. So the spongocoel is simply the space inside the sponge, or the cavity inside the sponge, and then the water is pumped out the top of the sponge through an excurrent pore. And there may be, in different species of sponges, there may be many excurrent pores, but a lot of times, or in a lot of species, there's one excurrent pore, and that's called the osculum. So the osculum is the term to refer to the one excurrent pore that the water goes out of in a sponge. You'll notice that there are these kind of hair-like things sticking out around the osculum. You can see them here too. And those are spicules. And they're kind of like a skeletal component. They're a supportive structure and also a protective structure. So spicules protect, like they keep small fish from swimming inside the sponge and maybe feeding on the cells lining the inside of the sponge, and other organisms from just climbing right in. But they also help to support the sponge because there are other kinds of spicules that are embedded throughout the cells of the sponge, or throughout the body of the sponge, to help support it. Okay, so sponges are the simplest kind of animals, and they're so simple that they're hardly more than a colony. But they are considered multicellular because they are made up of a number of different specialized cell types. And so that's what we're going to look at now. So if we zoom in on the body wall here, we see a number of different cell types. And one of the most important ones are the choanocytes. But if you're a very simple animal, the cells that make you up need to carry out many functions, a number of different functions. It's like that old saying that someone might wear many hats in a company. In other words, they perform many different jobs instead of specializing in only one task. So that's kind of the way the cells are in a sponge too. Each kind of, or a number of different cells are carrying out a number of different functions. So choanocytes are the ones that look just like choanoflagellates. They look very similar to choanoflagellates, and in choanoflagellates we actually call the individual cells choanocytes. So they have the same name because they are identical. So these cells are lining the inside of the sponge. So if we go back over here, it would be the inside of the sponge here that would be lined with choanocytes. And those choanocytes are there to set up feeding currents they have flagella, right? Choanoflagellates, they have flagella, and those flagella beat, and that's what moves the water through the sponge. That's what's pumping water through the sponge, and sponges can move a lot of water. So as the water is pumped through, food particles in the water get trapped in the collar of the choanocytes. So down here, notice that I labeled the collar of this choanocyte, and that collar has mucus. So food particles, when the food particles run into the collar, they stick in the mucus. And then the cell engulfs the food particle by phagocytosis and digests that food. Remember, there are lysosomes, so the food particle will be taken in by phagocytosis, form a food vacuole. Lysosomes will fuse with the food vacuole and empty their digestive enzymes and acids into that food vacuole. The food will be digested, releasing the nutrients, and then those nutrients will be used by that cell, but then excess nutrients will be picked up by these other kind of specialized cells known as amoebocytes. So before we move to the function of amoebocytes, let's go back and just summarize the function of the choanocytes. So they function in setting up water currents, first of all. They function in feeding, but they also function in that they will differentiate into sperm cells when it's time for sexual reproduction. So when it's time for spawning, usually that's a seasonal thing. All right, so those are choanocytes. Amoebocytes are arguably one of the most important cells in the sponge, cell types in the sponge. They're kind of like a basal cell type because they will differentiate into other cells. So they can become choanocytes, for example, and epidermal cells, and porocytes, and these ones called sclerocytes. So amoebocytes function in immunity, and by the way, they're called amoebocytes because they crawl around like amoebas. They use amoeboid movement to crawl around within the mesoglia of the sponge, which is like mesoglia means middle glue. It's not made up of cells. It's made up of collagen primarily, and so are you. So a lot of what holds your body together is collagen, and collagen is thought to have been, the evolution of collagen is thought to have been a major component of the move from unicellular to multicellular, or from colonial to multicellular, because it helps to hold cells together. So that's the mesoglia, literally is a middle glue holding the cells together of the sponge. But the amoebocytes, as I was explaining from the collar cells, will pick up nutrients from the collar cells and carry those nutrients around in the sponge and deliver those nutrients to other cells. So that's what I'm kind of showing here. This amoebocyte's delivering nutrients to this epidermal cell, for example. The epidermal cells are differentiated cells too, and they not only just make up the covering of the sponge, but they themselves can actually take in food particles from the outside of the sponge by phagocytosis. So you kind of get the idea here that, every cell doesn't have just one job. Most of the cells in a sponge have multiple jobs or multiple functions. So the amoebocytes, being amoeboid, they crawl around inside the sponge, and if they find something that's not supposed to be there, like a bacterial cell, for example, they will engulf it and eat it. And so that's how they function in immunity. And that's kind of the most primitive kind of immunity there is to have cells crawling around in your body that find and engulf foreign invading organisms. But part of our immunity does the same thing. We've got cells in our body called macrophages that do exactly the same thing that these amoebocytes do. They crawl around in your body by amoeboid movement, and when they find a bacterial cell or something that's not supposed to be in you, they will engulf it and eat it. Macrophages means big feeders. So again, what I just explained is how they function in circulation, amoebocytes. They also differentiate, like I said, into other cell types. And one of the cell types that they differentiate into are egg cells. All right, so choanocytes will differentiate into sperm cells, and amoebocytes can differentiate into egg cells for sexual reproduction. So amoebocytes are a really important basal cell type in sponges. I mentioned spicules before, and so here's a spicule that's embedded in the mesoglia of this sponge. And again, it's a supportive structure, kind of acts like an endoskeleton, but we don't really consider it a true endoskeleton, but it carries out the function of support anyway, and protection, as I was explaining here with these spicules surrounding the oscula. But the spicules, depending on the species of sponge, can be made out of one of two things. Calcium carbonate, that's CaCO3, calcium carbonate. That's the same thing that shells and coral is made out of, calcium carbonate. Or Si is silica. So these spicules, depending on species, can be made out of calcium carbonate or silica. And silica is basically sand. Sand is silica. So both of these compounds, well, calcium carbonate is a compound. Silica, I believe, would be elemental. But anyway, both of them are readily available in the ocean in aquatic environments, and that's where sponges live. There are freshwater sponges, but most of them are marine. Most of them are in salt water, in the oceans. And the spicules are built by these specialized cells known as sclerocytes. So the function of sclerocytes, they differentiate from amoebocytes. So amoebocytes will differentiate into sclerocytes, and the sclerocytes then will secrete the substance to build the spicules, whether it be calcium carbonate or silica. All right, here we're looking at the classification of Phylum Porifera, along with an evolutionary trend that we see in poriferans. And so, first of all, there are about 9,000 species of sponges. And again, most of them are marine. There are some that are freshwater. But they are classified into three classes. Class Calcarea, Class Hexantinella, and Class Demospongia. And they're classified both by the arrangement of their bodies and by the spicules that they create. So, for example, Class Calcarea has calcium carbonate spicules, those supportive structures that are embedded in the mesoglia of the sponge, right? Those are spicules. And so they're known as hard sponges or calcarea sponges. And then we have class Hexantinella, which are known as glass sponges because they have silica spicules. And I mentioned that silica is what sand is made out of, basically. And if you may already know that we use sand to make glass. Glass, we actually melt down sand to make glass. And so these sponges that make their spicules out of silica really are made of glass. They are glass sponges. And they're very delicate and beautiful too. They're used in Japan like decorations on the bride during Japanese weddings. And then we have class Demospongia, which have no spicules. And they have spongin fibers to support their bodies. And the spongin is a protein that is very spongy. It's elastic. It's very flexible. So these are the kind of sponges that you could wash your car with. For example, if you wash your car with either a calcarea sponge or a hexantinellid sponge, you would scratch the paint right off. But these Demospongia, these would be like the baths, what we would commonly call a bath sponge. And so they're just squishy and spongy, and, they are what you think of when you think of a sponge. Over here, we're looking at the evolutionary trend that I mentioned, and you'll notice that there is some connection here to the classification. So calcarea sponges tend to be simpler sponges, and what that means is their body wall is not highly folded. Whereas in what are being referred to here as advanced sponges, the body wall has folds in it. And we see that in both calcarea sponges and hexantinellid sponges, both hard sponges and glass sponges. And then in this last situation here with that corresponds with Demospongia, they are referred to as complex because they are not only highly folded, there are all kinds of channels throughout the body of the sponge too. And what all of this does is increase surface area. So here again, time and time again in biology, we see this concept of surface area coming back. And so what's going on here is by increasing folding in the body wall of the sponge, it increases surface area, which allows for more filter feeding. because all the body wall here is lined with choanocytes. And the more body wall you have in contact with the water, the more choanocytes you have in contact with the water, the more food you could eat, the more food you can capture, the more food you can filter. And that allows you to grow it larger in size, which allows you to have even more channels and more folding. So the idea is that sponges started out simple, and through evolution over time became more and more complex. And the selective pressure for that is to grow larger in size and be able to filter more food. Finally, we're looking at reproduction in sponges, and they can reproduce both sexually and asexually. So let's just take a look at asexually first. They can all reproduce asexually by fragmentation. And if you're a sponge farmer or fisherman, I don't know which you would call yourself actually, if you were in the business of raising sponges, when you go out to harvest your sponges, you don't keep them all. Some of them you're going to chop up and throw back in the water again. And what that does is they reproduce that way asexually. So all those little fragments of sponges will grow into whole new sponges again. And that's asexual reproduction by fragmentation. And sponges are really good at this. They're so good at this that you can take certain species of sponges and actually put them in a blender and blend them to the point where every cell has been separated from the other cells. And if you let that mixture sit there, the cells will come back together again and reform into the sponge, believe it or not. They're also really good at regeneration. And where that comes in handy is, like sea turtles feed on sponges. And so the sea turtle will come along and it'll eat a lot of the sponge away, but then it'll leave some behind, and what it has left behind will regenerate back into the sponge again. And that sea turtle can come back and feed on that sponge again someday. So they have great asexual reproduction in the form of fragmentation, and they have great powers of regeneration. Actually, amazing powers of regeneration. As I mentioned, there are freshwater sponges, and some freshwater sponges can also asexually reproduce with these little things called gemmules. And not to be confused with gem A. If you'll recall back when we were studying liverworts, liverworts can reproduce with gem A. These are gemmules, and they're just little packets of cells. Little packets of cells that can overwinter, for example, and that's one reason for these freshwater sponges producing them. And then in the spring, they will grow into fully functioning sponges again. So asexual reproduction with gemmules. You already know a little bit about their sexual reproduction. One sponge will release choanocyte sperm, and I'm calling it choanocyte sperm just to remind you that choanocytes differentiate into the sperm. So one sponge will release choanocyte sperm, and that sperm will swim through the water, and just hopefully the sponges are close enough together that the sperm can find another sponge that has produced amoebocyte eggs. And the sperm, and I'm calling that an amoebocyte egg because just to remind you that amoebocytes differentiate into eggs. And of course, the sperm will swim into the body of the sponge and find the eggs and fertilize the eggs. And then the egg that will form a zygote, of course. I didn't label zygote on here, but that will produce a zygote. The zygote then will start to divide by mitosis and form an embryo. The embryo then will develop into a larva, and the larva will be released, and it has cilia, so it's a swimming larva, and it will find a suitable place to settle down and attach and grow into a whole functioning sponge that will be, a sessile filter feeder. So that's sexual reproduction in sponges. Very straightforward. And what you would expect. So that's it for sponges. That's it for the phylum Porifera. Next time we'll take a look at the phylum Cnidaria, the next evolutionary step.